Welcome, everyone. I'd like to thank the ACD for such a tremendous invitation to a great meeting here. And what I'd like to do over the next, say, 20 or 30 minutes is take a step back and talk a little bit about the process of developing a new therapeutic. And that process is guided by regulatory authorities. Here in the US, it's called the Food and Drug Administration. So what I hope to do is walk through that process. There will be lots of terms that seem a bit unusual, but hopefully I'll be able to explain what these different steps are. And I'll take a couple of different occasions, hopefully, and ground that in the efforts that are ongoing in the community to create a new treatment for CTD. Okay? Now I got to figure this out. This may be the hardest part of my talk today. How do we move it forward? Green arrow. Got it. Okay. I said the hardest part. So therapeutic development, what is this? This is a concerted effort by usually quite a large teams to develop an intervention, and it could be of different types, I'll talk about that is therapeutically beneficial for the treatment of human diseases, right? And so in this instance, it's one of the CCDSs, right? It is a long process. In general, and there are not a lot of statistics, people have looked at this, it often takes about a decade from inception to the point of actually getting something approved in there. And that can vary widely, and sometimes it's shorter or longer, but that gives you some idea about the length of time it takes. And it is highly resource intensive, right? There are studies that say that for therapeutics for very large diseases can cost hundreds of millions of dollars to support that effort to create a new therapy. So this is not somewhere we tread lightly. All right? And it's a highly regulated process. The talk today will be about the Food and Drug Administration, but there are equivalent agencies, regulatory agencies in places like Europe. There it's the European Medicines Association or agency, or in Japan or elsewhere. But today my comments will focus here on the Food and Drug Administration. All right? There are three different types, major modalities of therapies that we work on. In the upper right there are the ones you're familiar with, the tablets or the capsules. These are what you've heard today are small molecules. These are when medicinal chemists in beakers create novel molecules that your body has never seen before, and we develop them into drugs. And these are the historical ones we've been working on for well over 100 years, and they have certain advantages. You can take them by mouth. They distribute widely around the body, and if you work particularly hard, you can get them to cross the blood-brain barrier and go into the brain, okay? But most of them do not do that, right? The blood-brain barrier there is sort of there to stop things like that from getting there, right? So we have to engineer them to do so. Over the last 20 or 30 years, we've been developing a lot of what are called antibodies or monoclonal antibodies. So these are actually proteins. They're made in bioreactors, in bioreactor cells, and they have to be injected. You can't take them orally, right? And they're developed because they can be very, very specific and very selective, and they're quite efficacious, right? And that's a whole nother class of molecules that we develop. And then lastly, you just saw Dr. Skelton talk to you about gene therapy, which is sort of a newer sort of basic modality of treatment. And we've been talking about that, and they're using viruses as sort of vehicles to bring and correct genes, all right? But I think my talk today is going to focus on small molecules. Why do we care? Well, I find about somebody like the FDA. Well, I find myself in Texas. This is a state with a tremendous independence streak. And so we might stand around and go, why do we need any bureaucrats up in Washington or in Maryland to tell us what we want to do here in Texas, right? But there was a reason why the Food and Drug Administration was doing it. And that was before this was set up, you could sell, and a shuckster could do just about anything. And there are many examples, and you could see just of what was essentially purported to be safe and effective in the treatment of human disease that led to tremendous problems. So historically, the FDA, which although it was originally started in 1906, it really started in 1938, right? And if you can see, the year before that, a particular elixir killed over 100 people because it had antifreeze in it, right? So in 1938 is the act that created the Food and Drug Administration. And what's important is that act said that all the FDA has to do is make sure things are safe. That was it. Just have to be safe if you're going to move them forward, right? And over time, that wasn't sufficient, right? 300 deaths after that act was because of another tainted material. We are all probably familiar with the story of thalidomide, because many people in this room were aware at that time, where a sleeping pill actually caused a significant number of birth defects. So at that point in time, laws were passed that then gave the Food and Drug Administration the ability not only to show that something's safe, but that people had to prove that it was efficacious or worthwhile as well as safe. Right? And so the regulatory landscape has evolved over time. Right? At that time, they also set up an advisory committee, and later on in the talk, you'll see why they set up that proposal, where experts come and advise the FDA to help determine whether something is efficacious or safe. And then all of you, when you go and pick up a medicine, you got a little package insert. This was also prescribed at some point in time. And then, although I won't talk about it today, one of the more recent acts enables what are called biosimilars or generics, right? And these are all things that are regulated by the Food and Drug Administration. So what is the process of developing a drug? Well, it usually starts in the laboratory. It starts with a good idea, and then you have to do what we call preclinical development. That's a lengthy process. I'll go into that a little bit more. But if you're able to convince yourself that you have something that might be therapeutic, you actually do one of your first interactions with the Food and Drug Administration, and you apply to test in human studies. And that's called an IND, or an investigational new drug application. I'll touch on that in a second. If you meet that hurdle, you then start clinical testing. Dr. Barshup's going to come after I and tell you a little bit more about clinical testing, but I will touch a little bit on what some of the phases are in trying to prove that something is safe and effective. And if you do that, you then apply to market that therapy through what's known as a new drug application, or an NDA. So I warned you, there's going to be some abbreviations, some new terms, but this is generally the process that you follow in order to get a new drug approved, in this case for the treatment of one of the CCDSs. So you start with discovery, all right? This one slide can encapsulate years and years of work, all right? And typically what you're trying to do, and remember I'm going to focus on small molecules, is target something. Usually it's a protein, and you want to modify that protein. So there are lots of different ways to do screening, to look for that particular chemical or to modify it, and then you go through multiple iterations of trying to optimize it. Right? And you've heard a couple of different talks today about that. And our colleague Dr. Nadasan and the crew at Ultragenics has a particular target. They're trying to come up with something that will deliver more creatine to the brain. So they're working around that thing, and they're creating analogs, and you heard him talk about scaffolds and different chemistries that they're working on to try to find the best thing to move forward. Because it's very expensive. You can't move 25 things forward. They're going to have to pick one thing. They're going to have to optimize it to take it forward into human testing. Right? On the right-hand side of the slide is another major thing that we do, and this is, for any small molecule drug, it has to get to where it's intended to work. So we spend a lot of time using animals like mice and rats and using different, what we call routes of administration. We administer it orally or intravenously by vein, and we see how much is distributed around the body and where it goes. That's called pharmacokinetics, and we spend a lot of time doing that, right? We also spend time understanding if it gets to where it does, and when it gets there, whether it does anything, whether it modulates that protein, or pharmacodynamics. So you saw a slide showing that in a primate, they were able to achieve already early in this program that once they deliver it, they can get creatine into the brain. So they've been able to do some pharmacokinetic, pharmacodynamic experiments, right? And what's very important is what we term ADME. All right, another term here. What this is an abbreviation for is absorption, how it is absorbed by your body, distribution, where it goes in your body, metabolism, how do we get rid of it? Usually it's through the liver or in feces, etc., but how it's metabolized, and then finally excreted, out of the body. So we spend a significant amount of time understanding those properties of any potential new drug. Right? We go so far sometimes as to radioactively label a small molecule and give it to a mouse and then find out where it goes and quantitate all of that. Right? So once again, a significant amount of effort usually takes multiple years and a lot of time and effort, and usually a very talented team to try to identify that molecule you want to move forward. Right? Let's say you achieve that. Oftentimes, in our vernacular, you will have nominated what we call a development candidate, something that you think has all the characteristics to be a drug, and you want to put more time and effort into it, right? Well, in order to get there, you have to make it, right? And I can't tell you how many times over my career it's been a problem where we can't make the drug, right? Now, you might think that's simple, but this is a very complicated process, all right? In the FDA terms, it's called Chemistry, Manufacturing, and Control, or CMC. And what they require is you have to demonstrate to them that you can reliably make it in a highly purified form, and that every time you try to make it, it's the same. And that not only that, once you make it, it is stable. So you saw in a talk the other day, Dr. Balestrini was working on a compound that degraded pretty quickly. And obviously he wants to optimize it where that can't be the case, because that can't be a pill, because you can't have a pill that you have to go to the pharmacy every single day and take it in 20 minutes. It's got to be stable and sit around for months, either in the refrigerator or on your dresser, right? So there are lots and lots that go into this sort of CMC component of that. And the criteria, when you first start human testing, change over time. And by the time you actually get approved, it becomes even stricter and more rigorous, right? So this is a very important component to this, and historically, it's a source of a significant amount of delay. So to give you a sideline, as many of you know, Lumos is working on an analog of creatine called Cyclo Creatine. And as my colleagues at the time tried to increase the size of the synthesis to go from a very small amount, which was used in animals, to a much larger amount, the process included what we call an exothermic step, which means it generated a profound amount of heat, which means things can explode, right? Not good. So we had to spend time revising the way it was made. And now we actually have a process that excludes that step and can actually be made. So to give another example on that, we actually became aware that over-the-counter, not FDA regulated per se, or not heavily regulated enough, was companies trying to sell Cyclo Creatine. And we said, how could they do that? Because we know if you make a large amount of it, it's going to go boom, right? So we tested it. No Cyclo Creatine in those products. So you've got to be exceedingly wary, right? There's an advantage of going through the Food and Drug Administration process, because you know that you're going to get something that's reliable and pure on the other end, right? And so there's a cautionary tale in that, right? That's very relevant for this community, right? So what's the next set of things? So we've made the compound. We think we have the thing that's going to work. Now we've got to show, as best we can, that it's safe. How do we do that? Well, we do what are called safety pharmacology studies. These are usually done when we administer to an animal or in a test system either a dose or an amount that we think is around the therapeutic dose or slightly higher. And there's a couple of key areas we look at. One is the heart. Drugs, small molecule drugs, can cause problems with the heart. So we test that in animals and in porcine strips in various ways to make sure we don't alter the conduction of the heart. We make sure it doesn't affect lung function. There's even a test called the Irwin test where we make sure it doesn't adversely affect brain function, all right? So these are very important tools that you have to supply data like this to the Food and Drug Administration as support to go into humans, to help ensure that when we actually test it in humans, it's safe, all right? And so this also, although it's not a huge time-consuming process, does require resources, and many of the things that we thought were good before fail here. And then we got to go back and say, oh, it's not good enough, and that's why when we do these earlier things, we usually have multiple what we call chemical scaffolds, because if there's one of them that runs into a problem here, we go back and say, well, let's go to class B or class C. So it's a very iterative process. It takes a significant amount of time to get through to ensure safety, all right? Another big part of safety is what we call toxicology. So what is toxicology? It's a science in and of itself, but when it's used in drug discovery, what we're trying to do is we purposely administer very high doses. So this is above those doses that we think are going to work in humans, to animals, usually at least 2 species, to try to see what happens if you have too much. And the thought here is that we can get an understanding, if something goes wrong in those animals, what we can track in humans and what the safety may be, all right? So these can vary in length. They can be short, a couple of weeks. They can be long, a couple of months. They can be dedicated to things like making sure reproductive ability is not harmed, that things are not genotoxic or cause cancer, et cetera. So a significant amount of effort is done in animals to ensure the safety of a new molecule in humans, all right? And the importance, at least early on, is it helps us to determine, along with something we call allometric scaling, what the starting doses in the first phases of clinical studies are, all right? It also tells us about special safety monitoring, all right? So all drugs, let me pause here for a moment, all small molecule drugs have risks, right? So it's all about pharmacology. All the drugs that are approved today, that many of you in this room may have taken a pill this morning for high blood pressure or for some other ailment, the amount you've taken has been shown to be safe. But if you take too much of any of those, they are no longer safe. That is intrinsically true of almost all therapies, right? So understanding what way too much of it might be helps us to understand how to monitor or care for patients in early clinical trials. So for example, if we find in animal testing that the liver is affected, we can measure blood during phase 1 trials and phase 2 trials to make sure that the amount given to patients do not get close to those that affect the liver, right? And then lastly, how long you can treat in the early stages of clinic is dependent upon how much time you've spent in toxicology, right? So if we do a 28-day study, in that we can only treat in the early stages of clinical up to 28 days. And until we do those longer animal studies, can we then get the approval to treat humans longer than 28 days, right? So a very complicated process, but what's very interesting is you are now, after doing all the things I just talked about, ready to apply for an IND. But you know what I didn't mention? I didn't mention efficacy, right? So you saw today, over the last 2 days, that all of this starts with, well, does it work in an animal model? Is it efficacious? None of that is required to test a drug in humans. All you need is a strong rationale and prove that it's safe, and you can move forward, right? A lot of people don't appreciate that. Now, in our modern world, it's very hard to get the resources you need to go forward without showing, let's say, in an animal model that something works. But there are always, particularly in the neurosciences, a lot of trouble trying to understand, and you saw the struggle to whether a mouse does a certain thing in a particular experiment, whether that's actually going to translate to humans, right? You know, so it is not required, right? So when you apply for an IND, those are the main things: the pharmacology, those chemistries, and the clinical protocol is what they review. And it typically takes about 30 days, and they hand you what's at the end of that, either a study may proceed letter, which is usually a happy day, we can start clinical testing, or they put you on what's called a clinical hold, right? So the FDA, not only in the beginning, but through the entire process, has the ability at any time to say stop. And they can stop for any reason, and you have to go back to them and explain to them why you would want to continue for whatever reason. So it's another thing that you often hear, sometimes you may hear about, is that companies or people working on things were placed on a clinical hold. That's a regulatory way to slow things down to make sure that things are safe, right? So this all comes to the core concept here, and that is that in any instance in you're developing a therapeutic, you have to assess both the potential benefit or the real benefit when it's demonstrated against the potential risk or the real risk. And it's that balance that determines the ability to move forward. And one of the difficulties at the very beginning of clinical testing is you have no benefit. And all you have is potential risks that you outlined in all of those studies in animals, right? So it becomes very difficult, and you got to move very slowly and very cautiously to make sure that in your initial clinical studies, they're focused towards understanding safety so that you can then move into more and more people and then try to demonstrate benefit later on, okay? Clinical trials, right? As I said, Dr. Barship's going to speak about that now, but a couple points I want to make. One is There was a declaration made by a group of experts in 1964 called the Declaration of Helsinki. And this was very, very important because this was through a process, it's been amended a number of times, that's there to ensure that patients or subjects in clinical trials, that it's done in an ethical way and that they have been able to be fully informed of that and have signed and given consent. Right? So not only is the Food and Drug Administration a regulatory level, but even locally, as you start clinical trials, as you work with some of the clinicians in this room, they have applied locally to what are called institutional review boards, which is another level of check to make sure that those consents are written in the right way and that anyone who participates in a clinical trial is aware of what the issues are. Right? So multiple layers of safeguarding that make clinical trials remarkably safe to participate in. Now, what are the phases of development? Well, phase 1, when you really start, is done either in healthy volunteers or sometimes it's done directly in patients. They're usually small studies, a couple of dozen patients, and we test multiple drug levels because we don't know exactly which one might be active. And after you go through an IND, oftentimes they ask you to purposely start at a level that's below what we think may work, right? So those first studies are usually very short. And one of the challenges for anyone participating in a phase 1 study is they usually cannot confer benefit, right? They're too short in time, or the dose may be too low. So you're signing up for something that may or may not have risk, but you're not likely to get benefit from that, right? So think about the challenges inherent in that proposition, right? If you're successful in phase 1, and a lot of the things we did in animals like that, those pharmacokinetics and stuff, we measure all of that and we make sure that it's safe and it gets to where it wants to get to. You move to the second phase of testing where we try then not only to continue to characterize safety, but to try to understand whether the drug could work or not, okay? Those trials are often have what's called a placebo control, where patients, you come in, that you don't know whether you're receiving drug or something that looks like drug but doesn't have the drug in it. And that's an essential trial design to try to understand whether something could work or not, all right? And then phase 3 get progressively larger, where you're actually trying to generate enough data to support an approval, right? So what does it take to get approved? Well, it's very interesting. The FDA mandates that you require substantial efficacy, but they don't say exactly what that is. This is the exact terminology here. If you look at this, evidence consisting of adequate and well-controlled investigations, clinical trials, by experts qualified by scientific training and experience to evaluate the effectiveness of the drug, right? So they don't say you need 2 trials or 3 trials or of this size. Every single drug effort is different, and it really depends on, once again, that risk versus that benefit, and also what disease you're trying to treat, all right? But what you are required to do is show substantial evidence of efficacy, and that has to outweigh any potential risks. And if you achieve that, you can get approval to market that drug, all right? So it goes through a process, right? And once you submit to that process, that's the one I talked about with the NDA. There's another act called the Padula Act that set some timelines for the FDA, but they charged sponsors to do that. So if we're successful enough as a community, any one of us, ourselves, our colleagues at other companies, if we get to that point, we're going to have to write a check for $2.4 million for the privilege for them to review this material, okay? It'll probably be more then, right? But the thought is, the FDA is underfunded, and this is one of the ways, think about that, to actually pay the salaries so that they can review for new therapies, all right? If you submit the application, you got 60 days. They may kick it back to you and say, we don't even like it, reformat it, it doesn't look good. But if they accept it, it takes them 10 months to review it. So that's nearly 1 year in this process is just the final review from the FDA, okay? Once again, here, in order to be successful, the benefit has to outweigh the risk, right? And that's the fundamental component here. Right? Now, I described a pathway that is called a standard pathway. Now, if you're developing a new drug for hypertension or diabetes and millions of people are affected by that, you're doing very large studies, it takes a while. But there are alternative pathways through the Food and Drug Administration that you may have heard about. Things like fast track or accelerated or priority or breakthrough. And these are different pathways that have been created to try to speed up that review process, right? And I won't go into them in detail today, but happy to chat with anyone about them, later this afternoon. But the one I think we should talk about is something called the Orphan Disease Designation. So there was an act of Congress in 1983 that created the Orphan Disease Designation. And in order to qualify for that, the disease that you're studying has to have less than 200,000 patients in the United States. So looking around this room and seeing that we are in the orphan disease space, right? Yes, okay, And the second thing is you have to have a scientific rationale. So not only does it have to be rare, but what you're proposing to get that designation has to pass some muster to say, hey, this could potentially work, right? And now remember, this is not data in humans, this is earlier on, right? But you can then apply for that. And why do we apply for that? Why do we try to get that? Well, one is exclusivity. So this has been important, right? Because if without orphan disease designation, you can only move molecules into the clinic that you have patent life on, that you have some patent protection around that allows you then to recoup your thing and make profits on the other end. But with this designation, even if a molecule no longer has a patent life, you're guaranteed a certain number of years after approval where only you can market that drug. So that's an exceedingly important component of that designation. And then lastly, it can speed up some of the reviews and you get to waive some of the fees, et cetera, with that. So if you're working in an orphan disease, almost everyone tries to get orphan disease designation and use that to help navigate through the Food and Drug Administration. So it gives you an example, and this is a little bit depressing, but it gives you an example of what the outcome of this is. So this is a couple years old, but you can see on average, there's only about 30 or 40 new drugs that are approved every year, right? So not many make it through. And on the right, you can see what the success is here. And this is what's so difficult, right? So when you're in a preclinical setting, when you saw some of the work that we've all done, there's only about a 3% chance that something's going to make it to market. Right? And this is overall everything. Sometimes it's slightly better averages in orphan diseases, but overall, and even when you get to phase 3, you only got a coin flip as to whether that's going to be successful and you're going to get approved, right? So you can see that this is quite a big challenge. It's a difficult but very worthy endeavor to try to develop new therapies for human disease. So I think in summary, the biggest thing I can say is you guys can make a difference. You can participate in clinical trials, right? Either yourselves, your children, they're safe. There's a tremendous amount of work we do to ensure that people who participate in clinical trials do not get harmed. And you can personally make a difference and help not only the other individuals in the room working on this to advance a new therapy for CCDS. That's probably my most relevant message, all right? A couple of acknowledgments. First, I want to start off by saying Dr. Barshop, I also reside in San Diego. He used the sunset slide yesterday, so I could no longer use the sunset slide. So, I chose our B option, which is surfing here at Scripps. And if you look closely, a number of you know a friend to the CCDS community this is Rick Hawkins, the CEO and founder of Lumos, is just about to work his way into this. So that's why I chose this one. But I would like to thank a bunch of people. First of all, my colleagues at Lumos Pharmos, who have been dedicated to the development of Cyclo Creatine for CTD. I see the CTD research community. We couldn't do it without the basic, discoveries that really guide how we do drug discovery. I want to acknowledge the Vigilance study teams, not only the PIs, but everyone working on the Vigilance study, which is an observational study to support this. And then most importantly is the parents, the caregivers, and the patients with the different CCDSs. You're the reason why we're here, so thank you.
Thanks, Dave. I enjoy your talk very much. I have 2 questions along with one small specific information here. Let me see. So for my field of research, stroke, I know in Japan, they have a drug, anti-reactive oxygen species drug, available. So they did a clinical trial in Japan, successful. And then actually from Japan, from 2001, they already doctor had used that to treat a stroke patient within the first 24 hours. So that's in Japan. But actually, because that drug is so old, already past the patent period. So actually in America, no doctor can use that drug to treat a stroke patient, although we know it's working in Japan. In Europe, that's doing the safety trial right now. And now my question, two questions here. Number one, analysis. So in this case, if one drug already been approved by the equivalent FDA agency in other respectable country, like Japan, okay, in America in the system, is there any expedite process to do that? Number one. Number two, clinical trial is so expensive for the drug, something like this, the available, because no pharmaceutical company want to do that because the patent period already passed. Can NIH play any role to facilitate this kind of non-profitable drug clinical trial?
Okay. So the first question is, each regulatory agency is independent. So if you go to the FDA and you bring them a drug that may have been approved elsewhere, they say, thank you very much, that's very nice and good, but we're the FDA. So for the first part is, in general, that it's approved elsewhere is not sufficient in and of itself to get an FDA approval, right? But you have an advantage there, and that is you often have clinical data, safety data, et cetera, that you can bring in, they can look at, and it can accelerate the development within the United States. So depending upon the individual program, you usually have to conduct one of two things. Either you have to prove that the molecule you're making is equivalent to the one that's approved elsewhere, right? So that's one of the things you have to show, is that whatever you're creating is similar to that for them to even look at that data. And then generally you have to conduct at least one clinical trial they favor a US population because there may be ethnic differences in the way molecules distribute or different. So usually you have to do at least some work in the United States but that's a major advantage when you know that a drug is safe, is effective, and has been given to people outside of the United States, all right? But the real hindrance there is related to the economics of it. You mentioned it is usually those drugs, and there's a whole class of something called repositioning, where we're trying to take drugs that may have been approved for one indication that may work in another one, no longer have patent life. So if you're unable to get an orphan designation, if you're not trying to reposition or move that into a small population, there's usually nobody willing to take that on, right, because of exactly that issue, right? Now, I'm aware, I know the NIH has a specific program where they're interested in repositioning drugs, so that's somewhere to seek some guidance. And I have a couple of NIH colleagues here in the room, including one of the directors of the Institute, the clinical director. Please feel free to chat them. They may be aware of additional resources at the NIH to do that. But in general, that's a wonderful way to take a drug that we call de-risked and try to bring it into the U.S. and use an effective drug in the U.S. population.
Some people might know, but where are we at with the Cyclo Creatine in the process?
Yep. So as many of you have been following Cyclo Creatine, we've been working on that for quite some time. Right now, we are still in the preclinical stages of that, meaning we have not yet given it to a CTD patient. We haven't started that sort of phase 1 work in a CTD patient. And right now, we're doing both sides of that equation. We are trying to figure out exactly what the benefit may be as best we can in animals and what the risks may be to make a decision about moving into human clinical testing. And I can reassure you we will only do so if it's safe to do so. And we're going to have a real insight into that. We're going to be able to come back to this community and let you know where we are in the first quarter of next year. So we're not too far away from letting everyone know what the exact status of our efforts are.
Dr. Weiner, I enjoyed your talk, except the last slide. So, tell me about focus groups and how focus groups and patient advocates can help in the drug development process.
Yeah, so many layers. Now, particularly in rare disease. So the first and foremost is awareness, right? And you saw that earlier today. You may be aware, we're in here, but raising the awareness of the disease, making sure that there are screening efforts so that more patients who may have that disease or become aware of it, know of it, may receive treatment or may participate in clinical trials, right? Fostering, welcoming, companies and paving a path for us to come here. So initially, I think it was probably just Lumos, but now I see colleagues from Ultragenyx, I see colleagues from Genana. So creating an environment where sponsors want to come in and work on that disease because there's now a framework that makes it feasible to do so, right? And then lastly is, the decisions made by the Food and Drug Administration are heavily influenced by the nature of what the benefit may be, right? And advocacy groups are quite an effective means for them to understand from the patients themselves, whether the effects seen in clinical trial are meaningful or not, right? So you can, I'm hoping that this group gets the opportunity with one of the sponsors when we get there to actually be able to get in front of the FDA and educate them about whether that intervention creates a meaningful benefit for patients with, be it CTD or any of the CCDSs.
Going back to the FDA, I know in the GAMT community, we are all getting our creatine and ornithine from different sources. I was told through Texas Children's that I could not just walk into GNC. Obviously, the creatine is not FDA approved through GNC. They can pretty much say whatever they want to say, correct? So how do we know the difference of the effectiveness of the creatine and the ornithine products sold over the counter versus where I'm getting mine from that are supposedly pharmaceutical grade?
Right.
And getting into the brain and
Yep.
and doing what they're supposed to do.
Yeah, so I would urge the same caution. Anything that's done over the counter is suspect, right? And this was a perfect example with Cyclo Creatine here, even with creatine, because as all of you know, bodybuilders, you can walk into GNC, and if you're away, they may be very pure, I don't know, but I would be suspect if you're using them in a medicinal usage. Okay. Now I went through a process that requires essentially, FDA approval throughout all that, but there are other layers of things. So in other words, manufacturers who want to manufacture, be it creatine, can apply through other means so that their facilities are inspected and that their process and what they're producing is pure. That creates another level of assurance that what you're administering to your child in a therapeutic means is pure, and those are much, much better sources than anything over the counter, okay? And, there are different levels of regulation there, but once you've gotten to that, sort of a certified manufacturer of that and has gone through that process, and you can ask the manufacturer of that, and they're usually a couple, they have to continue, not only initially show, but continually show that they're producing a highly pure version of it. So those are much, much more preferred. Okay, and many times, a lot of those facilities have been inspected by the FDA themselves, right? So there's many reasons why, and it may cost more, and there's other things, but there's a major advantage there, right? To give you an example, a lot of times the FDA requirements for those are greater than 99% pure, right? So you can just imagine how much better that may be than another product that doesn't undergo the same scrutiny. Good question.
I guess my question would be, how come the costs can vary for the same thing from different places? I mean, I'm talking enormous amounts.
Yeah. So, difficult question, and I'm not going to be one to stand up here and defend an industry that struggles with that, right? I work on the research and development side, and I firmly believe, given the efforts, that there should be an incentive or some sort of we should have a system whereby you're rewarded for going through this tremendous effort and developing a therapeutic for human diseases. But I also, and this is me personally, feel that as an industry, we're abusing that, right? And that indeed, because of a lack of particular constraints, therapies can vary widely in their prices, and companies raise prices, and this is coming to a head currently over there. So that's a whole separate side of the thing that I don't really understand well. I don't work in that side, but I probably share your concern there, that we've got to do something to fix that, because at the end of the day, we have to maintain the ability to incentivize for profitable companies to want to go down this endeavor, right? But we also want to make sure that everyone who needs a drug can afford it and get it. So,
I have a question too. So how does population size influence the application process for the FDA, right? Like we're a rare disease, we're a relatively small community.
Yep. So it starts with the clinical trials. So if I were to tell you that I worked in Parkinson's disease and my trial I conducted 4 clinical trials all at the same time, and they had 600 people each in them, right? We can't do that. Right? So just out of practicality in rare disease, as you interact with them and we share with them the numbers, we have to combinedly agree what is feasible and doable. So they don't look at us and say, you must teach 600 patients, because there may not be that many in the entire United States, certainly not that we're aware of. So the size of that clinical program is usually fit based on the size of the patient population. Okay? Because in rare disease, there's often a lack of knowledge, right? So if I were to tell you about what a meaningful effect in lowering blood pressure is, we can have hours of talks about that. But what a meaningful effect for improvement in, say, CTD might be is up for debate, right? And so oftentimes in rare diseases, the trials are not designed to meet what we call statistical significance, right? We do trials. They should be rigorous. They should probably be placebo-controlled in most instances. But when you look at that data, you've got a little bit more leeway to look at the totality of that data and try to understand whether there's a benefit or not. And this is why coming back to parents through the advocacy agencies make it so important. Because the real arbitrators there, and the FDA will agree with this, is not the reviewers at the FDA. They want to know whether parents believe that this is a beneficial therapy or not, right? So things are scaled down and you're given a little bit more leeway with respect to what constitutes substantial evidence of efficacy, right? But one last point is the molecules still need to be safe, right? They are unwilling to approve things that raise safety risks, right?
So that was a great answer, but I think I have a follow-up too.
Please.
What about the potential market, right? So does it matter that, we have only, I don't know, like 3 potential patients kind of thing versus, you have the second cause for intellectual disability, or does that help in any way, right?
So once again, more on the business side, but you're correct. In order to make this entire endeavor economically feasible, those therapies usually are much more expensive than ones for broader diseases, right? And some examples of them ridiculously so, right? Gene therapies that cost $1 million a simple time, or therapies that cost hundreds of thousands of dollars. So in general, because the diseases are rare, healthcare systems and insurers can afford that because very few people have that, right? And oftentimes the rare diseases themselves cause enough expenses or incur enough expenses in caring for patients that you save money on that the economics of that can work, okay? But I also put out another, and this is another personal cautionary measure, that area of our industry too is going to receive a lot of scrutiny because I think it's been overused. Let me be nice about it in the way that that's approached, right? So there will be significant revision there in my mind in the future. Right?
I have a question about GAMP treatment. A lot of us struggle to get coverage for our supplements. And like Jennifer mentioned, we wonder what we're giving our kids. We bounce around between who's supplying it, who's run out of it, who's turned gritty and lumpy, and who's got a smooth creatine. And, it's kind of scary. But I've also heard people talk about you don't want an FDA-approved drug for GAMP because it's so messy and sticky. But as a parent, and there are some parents who are getting their drugs compounded so that their kids aren't gagging and it tastes good. But if you live in a state where you struggle with that, you just have to go to water and, or flavorings. Anyways, and this might be for other people in the room as well. Would you look at GAMP treatment as something that should go towards an FDA-approved treatment, or is that a major con to get it FDA approved, and we should just keep doing this ourselves, measuring powders and...
So, my advice, I don't think anyone is going to bring that forward for FDA approval. There's probably not a strong rationale to do that, nor would I advise the advocacy group or the community to do that per se. As a dietary supplement, as a natural product, right, if we can, and is put in place, ensure the quality of the product, like we had mentioned before, as long as there are manufacturers in that place that can manufacture at a very high level, I don't think the FDA process or getting that is going to change that, right, as long as you have that in place, all right? I think the more problematic thing in our healthcare system is what you mentioned before, is how those treatments are paid for, right? And so the big challenges, and I see it with colleagues who are clinicians here, is they spend a significant amount of time arguing with insurance companies trying to get reimbursements for that. So I think that's what I would focus is the challenge on that side is showing, these insurance companies that the benefits of creatine supplementation, and then working with the manufacturers to try to make sure that you get a steady source and supply of very high quality creatine. But I would not encourage, going through a formal regulatory process. It's not going to create any additional advantages.
So I have a question, kind of a follow-up question to that. My son with GAMP currently takes cytotine from Solace Nutrition. I think there's a bottle of it out there. It's orange. We just looked it up and it says they have the NSF designation.
So do you know anything about that? And is that similar to what you were speaking on before about
Yeah,
it being
tho-
safety
those
tested?
those are the kinds of things. I don't know about that one specifically. It's a little bit out of my realm. But whether it's NSF or whether it's some other, there are different, once again, not the Food and Drug Administration that do this. But I think the key importance, I'm going to head it over, is you really should speak to your clinician, because they know a lot about that and they can really inform you. And I will hand it over to an excellent one, Dr. Barsha.
No, I just wanted to start now. No, actually, I believe that cytotine is one of the products as a designated medical food. And I think that's an important designation. And it is possible then to get an insurance company to pay for that. But I hate to say this, but the way to get them is not to motivate them how much good it is, but to motivate them how much trouble they could have for not providing a proper product. So what we really need is some legal action for denying this and other proper products.
So I just wanted to say mine is covered, and I have a letter from Blue Cross and Blue Shield saying that it's covered as a medical food until he's like 23 or 26. So it is possible, but you do have to fight for it.
So if for GAMT we're going for the health supplements route, right? It strikes me that for creatine uptake, there's a huge health supplement market of, the CRT, the creatine transporter is very inefficient. Has what, like 10% uptake or something like that? So the health supplement, this perhaps very large market of health supplements would be very excited to find something that boosts the creatine transporter. So I would be happy with a health supplement.
No, we actually, and I've faced that questions multiple times. So as we were developing an analog of that is, well, are you interested in CTD, but eventually are you just going to go out there and, and sell it to bodybuilders and do all of that? At the end of the day, we're here to treat human diseases, right? That's our singular and sole motivation. So although you are correct, it certainly will not be me who's involved in such a process. That's all I can tell you.
Thank you.
Okay,
Thank you.
Thank you very much.
I'm sorry, I had a quick one. So I totally understand the economics where we got a small potential patient database and huge expenses to get there. So assuming this drug's going to cost, whatever, thousands and thousands of dollars a year, once the approval has been made, do insurance companies immediately just flip the switch and say, great, we pay for it? And I'm assuming it's no. What can we do kind of in parallel with the testing so that process happens quickly once it's hopefully approved?
I love the last part of your question. So yeah, the answer is no. The answer is no. So what we as sponsors often have to do is, once we get an approval, then things shift. Then people actually go and sit and present that data to insurance agencies and argue that the big insurers should cover that particular product. So they literally go one by one to all of these things and make what we call a health economic argument that this is advantageous for that insurer, meaning that, without this therapy, you spend X amount of money per year in hospitalizations and therapies on this patient, but with this therapy, it'll work out for you, right? So there's still a challenge. Even after a drug is approved, that does not mean an insurance company has to pay for it in the United States, and you have to make that argument, all right? But the more important part of your question is, what can you do or this community do? And the answer is, when sponsors that are working on this now get a little further along, you can help understand what that dynamic is. We can do things like health economic surveys. We can understand what the burden of CTD is, because it's hard to quantitate sometimes. And that information will, with whoever sponsor gets there, will be invaluable to making sure that whatever new therapy is covered by all insurers and that all patients get access. So I look forward over the ensuing few years to work to define that even further, but more coming. I think right now, focus on disease awareness, focus on newborn screening, identify this, but as different sponsors move along and get closer to that, we'll have that conversation, and you can participate and help make sure that any therapy that's approved is widely used by the entire community.
Okay, so you mentioned exclusivity. Sorry, I have lots of questions.
It's a very good session.
And I hold the microphone, so I get to ask it. So you mentioned exclusivity, and I assume that applies to the molecule, right? It's the exclusivity for the particular molecule, but the orphan drug designation does not apply to the only treatment for
No,
CTD, correct?
it's for the indication, for the molecule in that indication. So I could have orphan disease indication for Cyclo Creatine in CTD, which Lumos does have. Somebody else could take that and apply it for a different orphan disease if they thought it was a. So it's specifically exclusivity to be able to market that molecule for that disease for a certain period of time.
Wonderful. So that means, are there orphan diseases where you have more than one treatment available, right? Like, are the incentives compelling enough for all of you wonderful companies to have?
There certainly are. I know a number of the clinicians here who work in orphan diseases, particularly some of the ones, the earliest ones that Genentech and others went out. There are multiple versions for some of the replacement diseases. And yes, they all stand alone, meaning that either the orphan designation has run out for that prior one, or they've gotten, there's an open space to work in that space. So the answer is the orphan drug indication does not necessarily punitize or discourage people from coming in with a second or a third therapy. It doesn't necessarily do that, and I don't know of any instance where that's happened, but if there's usually first-generation therapies can be improved upon, and it's usually that the second or the third generations offer some clinical advantage, and market forces eventually bring that to the case.
Thank you.
Okay, you're very welcome.
Thank you.